Hi, I'm Colleen Cavanaugh. I'm a professor in the Department of Organismic and Evolutionary Biology at Harvard University and the co-director of the Microbial Sciences Initiative. Today, I'm going to tell you about this discovery of chemosynthetic symbioses. These are intimate associations between bacteria and marine invertebrates that allow both partners to colonize and thrive in otherwise inhospitable environments. The discovery of these symbioses followed on the discovery of the deep sea hydrothermal vents and the communities that flourish around these. But finding the symbioses at the vents also highlighted that these occur in other habitats, such as coastal sediments, that have a lot of sulfide. So today what I'm going to do is first provide you with some background on symbiosis, on chemosynthesis, the basis of these symbiotic associations, on the hydrothermal vents, and then on my academic path through this adventure. So symbiosis the term was coined by Anton de Berry to mean the living together of differently named organisms. These are associations with benefit to at least one of the partners. And as examples, there are corals, which have intracellular photosynthetic algae that feed them, legumes that form symbioses with nitrogen-fixing bacteria, so effectively they're self-fertilizing, chloroplasts and mitochondria in our cells in plants, which evolved from free-living bacteria. And of course, recently, we know about the human microbiome. There's been a lot of work in the last 7-8 years. And the impact that your own bacteria, those within your gut, on your skin, and all over your body, have on the development of your immune system, nutrition, neurobiology, the list goes on and on. So symbiosis is an incredibly important aspect of biology. So the common theme here for these symbioses, and one of the main processes, is CO2 fixation. Carbon fixation is typically, we think of it by plants, which is via photosynthesis. This is taking carbon dioxide out of the air and turning it into sugars. And in plants, you use light to fuel that operation. In bacteria that are capable of chemosynthesis, they use inorganic compounds such as hydrogen sulfide, iron, hydrogen, and oxidize these compounds to produce the energy that is then used to fix CO2 in the same processes as photosynthesis. Deep sea hydrothermal vents are ecosystems on the seafloor. They were discovered in 1977 in association with the search and study of seafloor spreading. Essentially, these are hot springs that are associated with volcanic activity along the ocean ridges. You can see in the top photograph that these ridges circle the Earth like seams on a baseball. These are areas where the seafloor is spreading. There's a lot of volcanic activity. Seawater percolates down into the deep ocean, or the subfloor, is heated, chemically charged with compounds, and then is forced up through other fissures, as you can see on the bottom photo with the so-called black smoker. And around these communities, around these vents, are these lush communities of tubeworms, clams, mussels, shrimp, with biomass that rivals that of the rainforest. This totally revolutionized our perceptions of life in the deep sea. Typically, it was thought to be very, very sparse because food from the surface, photosynthesis, wouldn't make it down. And so it was really, how is this working? Bringing up samples of seawater on some of the first cruises by geologists, they noted that it smelled very strongly of hydrogen sulfide. And it was proposed that bacteria using such compounds could be the base of the food chain here via chemosynthesis. As you'll see from the rest of the talk, in addition to free-living bacteria supporting these ecosystems, symbiosis is prevalent and, in fact, the dominant form of carbon fixation at the vents. So my path to chemosynthesis and symbiosis in the deep sea started at the University of Michigan. I'm from Detroit and went to Michigan. And there was a marine ecology course offered at the Marine Biological Lab by 3 Michigan professors. It was a Michigan course. We had 2 weeks of lectures and then had to design our own project. And to me, it was incredible that I learned that not everything is known, that textbooks aren't always right. And I fell in love with research, and I fell in love with Woods Hole. and managed to work out so that I could come back after graduating at Michigan. Then I worked at the Ecosystem Center with John Hobby, and during this time I was basically in a dark room looking in a microscope the whole day, all summer, counting bacteria. He had developed a method using fluorescence microscopy for actually being able to count bacteria in the real world that are too small to be able to be seen with a light microscope. So it was a very revolutionary period of time on numbers of bacteria in the world. I then went to Harvard to graduate school. My plan was to work on coastal and open ocean nutrient cycling, as this was part of the research that I was doing when I was in Woods Hole. So this brings me to Harvard. As a first-year graduate student, I was taking a class called Bio 255, Nature and Regulation of Marine Ecosystems. It was basically three professors that offered it. They talked about their own research, and then they brought people in to talk to the students. And because the vents had only been discovered recently, we had a series of talks on them. And the first was on the ecology by Ruth Turner, a professor in my department. The second was on chemistry by Russ McDuff, who was a chemist at MIT working on vents. The third was on microbiology. Holger Janisch was from Woods Hole, from the Woods Hole Oceanographic Institute. I knew him there. And he talked about the presumed microbiology, chemosynthesis being the base of the food chain, and a lot about sulfur bacteria, iron bacteria, and many others. Last, we had Meredith Jones, the curator of worms from the Smithsonian Institution. He came, and he basically was an histologist and a taxonomist. And in class, he described these tubeworms, seen in this picture, which can get up to over 1 meter in length. And it turns out, very peculiarly, they did not have a mouth or a gut. And so the big question was, how do they feed? And essentially, they're smaller relatives that could take up dissolved organic carbon across their epidermis, but these were so big it was thought the surface-to-volume ratio was much too small, and the concentration of organic carbon at these vents was low. So amazingly, he was basically describing the tubeworm section by section through a 1 meter tubeworm, and I was still awake when he came to this slide, and this is the so-called trophysome tissue. This is the tissue that fills the coelomic cavity of these tubeworms. It's a brown spongy tissue. It was named trophysome in previous descriptions because it has the gonads embedded in it, and the tissue was thought to feed the developing eggs and the developing sperm. So he showed this picture during the talk, and if you look in the back, that's a millimeter ruler, so this is a regular photograph. And he mentioned that he saw all these crystals, and he had them analyzed, and it turned out they were pure elemental sulfur. It was at this point where I jumped up and said, well, it's perfectly clear. They have a symbiotic association with these chemosynthetic bacteria that live inside of them, and then they feed the animal just like dinoflagellates, the algae and corals feed their corals. He said, no, we think it's a detoxifying organ. And I said, well, that's perfect, because these bacteria can oxidize sulfide to elemental sulfur and render it less toxic, or not toxic, and then still fix CO2 and feed the tubeworm. So after the talk, I was able to convince Dr. Jones to send me some tissue and that I would prove there were bacteria there. Indeed, I thought it was a done deal. I thought it was a given, but I actually had to jump through many hoops to try to prove that there were bacteria in this tissue. Now, I might mention I had never had a microbiology course, and I had worked for John Hobby almost well over a year plus, so I had a love of bacteria, but I was looking at the majority of which could never be cultured. So really jumping in at a basic level. I had done scanning electron microscopy, which is a method to be able to look at tissues at higher magnification. And you see on this higher upper figure, this is a scanning EM of the trophysome tissue, which is very lobular. And what I did was took a piece of tape and pulled off a little bit of the surface. And if you look closely, you'll see that it looks like the bottom figure is what it is. Basically, it looked like lots of grapes to me. Large type spheres. And I was so disappointed because I was used to looking at tiny little things that were bacteria, and I was like, oh my goodness, these are huge. Basically, the scale bar at the bottom is 10 microns, so they're about 2 or so, and I was used to looking at 0.4 micron bacteria. I talked with Holger Janisch at HUI about this, and he did point out that symbionts, let me show you. First, I did find a couple rods, so it was promising. A little, but only a couple. I then talked with Holger Janisch, and he explained that many times bacterial symbionts, when they're intracellular, they lose their cell wall because they're osmotically protected. And so they can have very weird shapes or even just balloon to spheres. So I had hope. In addition, we looked where the blue arrows show what look like delineations of the host cells, and that we inferred we were testing whether they were actually intracellular or not as well. So another line of evidence that I went after next was examining whether these spheres had DNA. And to do this, I used epifluorescence microscopy using a stain called DAPI. And this brings me back to my work with John Hobby. So I thought if these spheres had DNA, then I would have another line of evidence. And indeed, as you can see in my notebook, these are how I did all of my microscopy, which is by drawing. You can see these spheres, and I have notes that they're about 1 to 2 microns, and that they stain with DAPI, indicating that they have DNA. So this is another line of evidence. I also, to check the size, took a scraping of my cheek cell, which you see in the lower side of this notebook page, and it turns out that the nuclei were about 10 to 12 microns. So I had another line of evidence that these things, the grapes, were bacteria. But some of the more definitive work came with transmission electron microscopy. This is where you take thin sections through tissue, and it actually shows the ultrastructure of the internal organelles and organs. And indeed, these look like bacteria. So the bacteria are labeled B in this slice through the trophysome, and they have a typical size and morphology. And if you look at the panel on the right, they have the typical cell envelope of gram-negative bacteria, those inner 2 lines, and then an outer membrane that's inferred to be that of the host, and indicating that they are within the cytoplasm but separate. So taken together, all of these data, along with work by other researchers, which had shown that there are enzymes associated with chemosynthetic bacteria in the trophysome tissue, and other data, we concluded that this is a symbiosis between these giant tubeworms, and that they form associations with sulfur-oxidizing chemosynthetic bacteria. They have not yet been cultured. They fall within the gamma proteobacteria determined by DNA sequencing. And there has been lots of research on these, which I heartily suggest you look at. In general, they're shown to have many, many adaptations to this symbiosis, which include, of course, being mouthless and gutless. They're being fed internally, but also biochemically. That red you see in the plume is a hemoglobin, and it's unique in that it binds sulfide and oxygen in separate locations. Our hemochromate is poisoned by sulfide, but here they managed to deliver both compounds to the bacteria separately. So given this sort of symbiosis and the benefits accrued to both, it's a nutritional symbiosis. The worms get internal nutrition. They're able to live in areas that they don't need photosynthetic food. And the bacteria, which I have with my bacteria-centric point of view, benefit because they get compounds for energy generation from both anoxic and oxic environments. At vents, the sulfide is produced geothermally, but the oxygen is in the overlying seawater. And by colonizing an animal, coevolution, adaptations, the animal is bringing the sulfide and oxygen to these bacteria. Given the benefits for both, I would predict, and did, that these would be everywhere, not just at vents, but in sediments that also, like muds, that have sulfide. So you can imagine my delight when a paper was published in May, only 2 months after I started work on the tubeworms, entitled Gutless Bivalves. They must have chemosynthetic symbionts. They were mouthless and gutless, and they lived in sulfide-rich muds. And there was a congener in Woods Hole right off the coast. I had already planned to be in Woods Hole that summer working on another project, but I completely switched and moved down, and I had another fortuitous event, and that is that Holger Janish's microbial diversity course at the Marine Biological Lab was focusing on sulfur because of the vents. And he had the sulfur microbiology dream team, which included, of course, Holger, Gijs Kuenen, Yehuda Cohen, and Jan Galchuk. And effectively, I moved in to learn about sulfur bacteria. And as you can imagine, I was trying to figure out how to find a 1 micron, 2 micron size bacterium inside of a 1.5 centimeter long animal. I didn't have elemental sulfur as a clue where to look. But fortunately, Gijs Kuenen pointed out that Rubisco, the CO2 fixing enzyme of the Calvin cycle, which is unique to plants and autotrophic bacteria, could be used as an assay for localizing what tissue bacteria might be in. And indeed, the gill tissue of these animals had very high activities of Rubisco, as you can see in this graph. And indeed, with transmission electron microscopy, was able to show that there are rod-shaped bacteria living within the gill epithelial cells. And the close-up shows that they have the typical gram-negative cell envelope, and these too are included in what appears to be a host membrane. Direct counts of these indicated there are 10 to the 9th to 10 to the 10th bacteria per gram of tissue. And they also have enzymes associated with sulfur metabolism and fixed CO2 depending on various sulfur compound additions. So we concluded indeed that this also was a symbiotic association with chemoautotrophic sulfur bacteria. Since then, we've done a lot of molecular characterization. They are gamma proteobacteria, and while there are strain level differences, it appears to be a monoculture. So again, a very specific symbiotic association. The discovery of chemosynthetic bacteria in the tissues of Solamaya also helps explain a rather peculiar behavioral aspect. They produce Y-shaped burrows and hang out right at the apex of the Y. This had been suggested to either be a refugium from predators, or perhaps they were gardening bacteria within the tubes. However, the symbiosis explains why they would do this. They're pumping oxygen from above, from the overlying seawater, and sulfide from below in the sulfide-rich sediments. So they're creating the habitat that the bacteria need and flowing sulfide and oxygen over their gills. The bacteria fix CO2, feed the animal is feeding the bacteria. So here we had two examples, one at the vents and one in reducing sediment, just coastal sediments. And this indicated to me, indeed, that these must be everywhere. And it turns out that all of the major macrofauna at the deep-sea hydrothermal vents have chemosynthetic symbionts. This includes the bivalves, the vent mussels, and the giant clams, and also shrimp, which instead of having intracellular bacteria actually have them on their surfaces, epibionts. Further, when I and many researchers around the world looked at other organisms that live in sulfide-rich muds, found these symbioses with oligochaetes, with nematodes, and with colonial ciliates. And indeed, the picture that looks like a rope, that is a nematode, and all of the filaments on top are actually the bacteria. So again, these are on the outside. So this hopefully demonstrated that these chemosynthetic symbioses are extremely widespread in nature. They occur in habitats at the interface of oxic and anoxic environments, such as the vents and sediments. And all I've told you about here is the discovery phase. There has been a lot more research continuing on these various symbioses, ranging from genome sequencing to looking at optimal growth at deep sea pressures and chemical gradients. And I'll just point out, as again, bacteriocentric, Given the remarkable metabolic breadth of microbes, I predict that there will be many, many other symbiotic associations, not just with chemosynthetic bacteria, but with others that have diverse metabolisms that would be advantageous to animals and other eukaryotes that cannot achieve these sorts of processes. And on that note, as I challenge my students, both in class and in the lab, expect a discovery. Thank you.